now I'm going to talk about pain modulation through endogenous mechanisms in our body, and also about drugs that help to block pain transmission and sensitization. Okay, so I drew this little figure here. So this blue neuron here represents projections from the periaqueductal gray matter and the medulla oblongata, and from the locus coeruleus, which is also, I think, connected to the periaqueductal gray matter, but they're in different places. And I think the locus coeruleus is in the midbrain, if I'm not mistaken, and then the raphe nucleus is in the medulla oblongata. The locus coeruleus has adrenergic norepinephrine neurons, secreting neurons, and the raphe nucleus up in the medulla oblongata has serotonergic neurons there. So these neurons come down the spine and go to different areas in the gray matter at different levels in our spine, and they can secrete, in the case of some of the ones from the periaqueductal gray matter, those can secrete endorphins, like endorphin beta, dynorphin, and enkephalin. Dynorphin binds to to kappa opioid receptors, and the other two, enkephalin and endorphin beta, bind to your mu and delta opioid receptors, which I'm illustrating as this red receptor here. And I'm illustrating the red receptor to represent multiple kinds of receptors, but I didn't want to draw every one of them out. So anyway, I just say that to say we have pain modulation mechanisms that use neuropeptides. Neuropeptides are actually come from DNA directly from proteins. So DNA gets translated into a pre-mRNA, and then some RNA processing happens there. Then you get this RNA that gets translated, and then those proteins also get spliced. So you can end up with multiple neuropeptides even from one DNA transcript. In fact, you can get different neuropeptides coming from the same DNA segment, and it gets protein spliced, which is very odd. But anyway, so we have neuropeptides that can modulate our pain, and norepinephrine and serotonin. Okay, and I'll talk a little bit more about that in a little bit, but I'm just going to talk a little bit about signal transduction here first. So this blue area here, I've shown the calcium channel, N-type calcium channel, and those, it starts out closed, but when you have an action potential, and this is the primary afferent neuron, for example, here. So when the primary afferent neuron fires, because you had like a signal of pain in the periphery that comes up, the signal goes through the dorsal root ganglion, where the cell body is for this neuron, and then goes to the end here. This could be Rexid's lamina 1, for example, or 5, but let's just say Rexid's lamina 1, because a lot of C fibers go there, I think. So in that case, you have depolarization. The depolarization triggers calcium channels to open and let calcium into the cell, and there's a lot of calcium outside and hardly any in the inside of the cell. When that calcium is released, that can trigger vesicle release. These vesicles, actually didn't draw any neurotransmitter on there, but these vesicles are packed with glutamate, mostly, in the case of these dorsal root neurons, or even in the brain, most neurotransmitters are actually glutamate. So what happens is these glutamate vesicles fuse right here with the terminal bulb membrane at the presynaptic part of the synapse. This is the synapse here, and then on the other side is the postsynaptic neuron, and this would be a glutamate receptor, for example, and the postsynaptic neuron. This postsynaptic neuron is actually your second order nociceptive fiber that goes up to the medulla oblongata, or actually, the ones that you actually feel discriminatively for pain sensation go up to the thalamus and synapse in either the posterior ventrolateral nuclei, or some of them synapse in more medial nuclei in the thalamus, in the posterior part of the thalamus. And those can send signals up to your brain, up to your primary somatosensory cortex and your secondary somatosensory cortex, where you actually perceive pain and can localize it. Some of these secondary, second order neurons go up to areas in your brain like eventually the cingulate cortex, the orbital frontal cortex, and the insula, and then in like your amygdala and your hypothalamus. This part, the periaqueductal gray matter, also, and kind of like in a feedback loop. So when the periaqueductal gray matter gets stimulated, whether it's from this directly or not, eventually the periaqueductal gray matter can send a signal down to modulate pain if that's favorable, if that's supposed to happen. So when this happens, you have glutamate coming over, binding to the AMPA receptor, and I can't remember what AMPA represents, but you also have other glutamate receptors, the N-methyl aspartate receptor, which I guess I could draw real quick. We'll draw it like a lighter green color here. So this is your NMDA receptor. And where's my black marker? Huh. Hold on a second. My black marker. Okay. So that's the NMDA receptor that brings in calcium when it's activated, but it has to be activated by two things, which I talked about in the last lecture. It has to be activated by both depolarization from like a lower voltage, which triggers an action potential, but this more positive voltage from the sodium that's coming in causes kind of depolarization even locally as an excitatory postsynaptic potential, and that can cause the NMDA receptor to open up and let in calcium. So I'm going to use the same color as that. Calcium comes in, and that calcium can signal things like, I don't know, we'll just put CAM K2, and that can do things. Eventually ERK gets activated, and ERK can cause these potassium channels to be more closed. It can also trigger other pathways that probably sensitize these sodium channels, so they're more likely to open to let sodium in to depolarize the neuron. Things like that. Oh, and also more AMPA receptors will be probably transcribed, or probably transcribed also, but at least put into the membrane by vesicles when they get phosphorylated. And so with more of these receptors, when even one of these vesicles opens up and lets a few out, it's more likely to activate this neuron at even lower frequencies than before. At a high frequency, you have a lot of glutamate coming out of the synapse, so that makes this neuron more sensitive. So that's kind of how the signal transduction process works. Delayed rectifier potassium channels, when they do open up, it causes, this is actually wrong, when glutamate comes in, it actually is more likely to close these potassium channels. No, actually, never mind what I just said. So these start out closed, for example. They're open at some frequency because they leak potassium through a little bit, but when you have signal transduction, these potassium channels help to repolarize the neuron, but they also have other functions in just gauging how much, how positive or how negative this neuron is. So when they're open like this, this neuron is much less likely to actually be depolarized because it has positive ions leaving, basically, and that helps it stay more negative. Because when you depolarize it, you're bringing it from a really negative or some negative charge to a more positive charge, and that changes the conformation of channels and other proteins that allow signal transduction. And the most prominent one would be the voltage-gated sodium channel, which does most of the conduction of signal. So I say all that just to tell you a little bit about signal transduction. So when say endorphin beta gets released from this neuron here that comes down from the periaqueductal gray matter, and it binds to this red receptor here, it triggers the opening of this potassium channel. So what it does is it allows this to happen here, and I guess implicitly then it's inhibiting that activity there. So when it does that, it causes hyperpolarization of this neuron, makes it less likely for this one to be activated because you need more sodium to come in and stuff like that. Whereas in this one, so when endorphin beta binds to this, it's inhibitory towards this calcium channel and causes it to not be as likely to open up. And if this calcium channel is not as likely to open up, then it's not as likely to cause the release of glutamate by allowing vesicles to fuse. So it doesn't let vesicles fuse as easily via calcium. So beta endorphin does this, enkephalin, basically this is representing the mu and delta receptors, and in the case of kappa, I believe it does the same thing. So dynorphin would also cause the same activity. And I also think that they inhibit adenylate cyclase in these neurons, which also is inhibitory towards neuroconduction. But then we also have other molecules like norepinephrine from the locus coeruleus. Norepinephrine binds to what's called the alpha-2 receptor, and the alpha-2 receptor is also found in both of these neurons, the presynaptic and the postsynaptic neurons in the dorsal horn. Norepinephrine agonizes the alpha-2 receptor, and the alpha-2 receptor in the body is kind of like a negative feedback loop to the sympathetic nervous system. You can find it in your pancreatic beta cells. I think, I'm just going to blank about the other places. I think it's in the hypothalamus, and it's probably all over the place in the body, but it's a negative feedback loop to the sympathetic nervous system. So in neurons, though, like this, it's going to also do the same thing. It's going to inhibit calcium coming through, and this is probably via, I think, a G-coupled receptor is what this is. So G proteins signal, and they result in calcium not coming in as readily. And on this postsynaptic neuron, it results in potassium being more likely to leak out to keep the neuron a little more hyperpolarized. So maybe it's at 90 millivolts instead, negative 90 millivolts instead of negative 70 or something like that. So that it makes it so more cations have to rush in for it to depolarize. So that's also what norepinephrine does. Serotonin, I don't know much about what serotonin does, but that comes from the RAF nucleus. At least that's one source of serotonin. And that can also have effects. You have various 5-HT receptors in your spinal or trigeminal spinal nucleus in your medulla oblongata, where your pain sensation comes from your face and from the trigeminal nerves. I know that sumatriptan, which is a 5-HT1 delta agonist and also a beta agonist, or 5-HT1D agonist and a 5-HT1B agonist, can affect not just your like vasoconstriction, like up in near your meninges, your blood vessels up there, that might be affecting your trigeminal nerves indirectly, but it can also affect in your trigeminal spinal nucleus. It might also be affecting this kind of thing here, where it's inhibiting, somehow inhibiting pain by being an agonist, like serotonin would be to the 5-HT1D receptor. Anyway, but in the dorsal horn, I'm not sure exactly about signaling, and also serotonin can be pro-nociceptive when you get bit from like a bug. I think they release serotonin, and that can cause, say, like vasoconstriction in your local blood vessels, which causes that toxin to even remain localized in your near the bite and just stay there. Can cause pain and stuff like that, which is kind of odd in the context of what I just said about pain sensitization. But I guess 5-HT there, the serotonin receptor in the periphery, can cause pain at least, and I think causes vasoconstriction. Anyway, moving on. So you have these, and then you have a GABA, I guess. GABA is another one that tends to do this through the GABA B receptor, which is a metabotropic receptor, and I think it uses G protein coupled signaling to open potassium channels, and I think it also opens or closes the calcium channels so you don't have the release of the vesicles. Let's see what else. Those are the main neurotransmitter signaling mediated inhibition of pain sensitization examples that I can think of. But then you also have other ways to inhibit pain transmission, and that's through inhibiting the sodium channel. So before, or I've also talked a little bit about it today. Sodium channels, when your neuron becomes depolarized, and I can't remember how many millivolts right now, but when it goes down to a certain, or should I say it goes up to a certain voltage, is not as negative, for example, it causes these sodium channels to activate and open up. When they open up, they let in more sodium, and that sodium also causes the same depolarization effect. So more cations are rushing in. Well, there are some drugs that can inhibit these sodium channels that are used for various things, like for seizures, because for a seizure you want to stop transmission, especially if it's really rapid and happening a lot. You want to inhibit it. Some antiarrhythmics, like class 1 antiarrhythmics, class 1A and B antiarrhythmics, cause inhibition of sodium channels by different ways. I think class 1 also inhibits the potassium channel, but in the case of class 1B antiarrhythmics, they are actually phasically inhibitive. So whenever the neuron, and I didn't tell you this today, but whenever the sodium channel opens up, it goes through a phase where it's less active, where it resets, but before it resets, it inactivates. And this inactivated state, it can't be activated again. Even if the voltage drops to the right level, it doesn't matter. It stays shut for a short period. Then it switches back to this state again where it's ready to open again. Well, in this state, it can be activated again by the lower voltage. But in the case of the inactive state, it can't. So drugs like lidocaine, which is a local anesthetic, which blocks pain, and is also a class 1B antiarrhythmic. In the case of, I'm trying to think of some of the other ones, oxcarbazepine and, oxcarbazepine and carbamazepine as the name of the drugs. Those drugs can phasically inhibit the sodium channels, and let's see, I know there's others and I'm just not thinking of them right now. Well, there's also like prilocaine, even cocaine does this. A lot of the canes that are local anesthetics block these sodium channels. Oh, tricyclic antidepressants can cross-react and also inhibit these channels as well a little bit. So if you're taking those kinds of antidepressants like amitriptyline, nortriptyline, imipramine, and the ones that sound like that, basically, those drugs can also inhibit these sodium channels, but I'm not sure if that's phasic inhibition or not. But they also have the other effect, too, of causing more release of norepinephrine from different neurons, too. That's why that's, according to the monoamine hypothesis of depression, that's part of the problem with being depressed is you're not getting enough serotonin and norepinephrine in your synapses. Well, tricyclic antidepressants inhibit the serotonin reuptake inhibitor, which causes, like in the case of other interneurons in the brain, causes the serotonin to be taken up faster. But inhibiting those serotonin reuptake inhibitors causes the serotonin to remain latent in the synapse for longer, so it can bind to the postsynaptic neuron. And in the case of norepinephrine, inhibiting NET, NET is the norepinephrine transporter. Inhibiting that allows more norepinephrine to remain in the synapse. That's what these antidepressants do, like tricyclic antidepressants. I think tricyclic antidepressants are older than some of the other ones. Even the serotonin norepinephrine reuptake inhibitors and the SSRIs, which are specific serotonin reuptake inhibitors. But so the older ones, or at least the tricyclic antidepressants, cross-react with even like histaminergic and cholinergic receptors, and then these sodium channels. So those actually have those two roles that I talked about that can inhibit pain transmission. The serotonin norepinephrine reuptake inhibitors like duloxetine, which is also Cymbalta, that has those two. It's also keeping both of those that come from these two places in the synapses. Serotonin and norepinephrine again, which can agonize receptors like the alpha-2 receptor, that clonidine, or also known, I think might be catapres, if I remember right, activates this receptor too, to promote potassium efflux in the postsynaptic and inhibit calcium influx in the presynaptic neuron to stop transmission. Also drugs like, gabapentin, which is a Neurontin, and pregabalin, which is Lyrica. Those drugs, even though they both have the word GABA in them, I don't know that they actually do anything with GABA signaling. Maybe they were designed for that, to be like a benzodiazepine or something. But they don't. They actually, because benzodiazepines cause a similar effect to this through the GABA-B metabotropic receptor. But those instead, they actually bind to subunits of the N-type calcium channel, and instead of the calcium channel even getting up to the membrane to be assembled, they would bind to one of the calcium subunits, and it never actually doesn't actually get to where it's going. It does that. It blocks it from even going up there. So those also have utility a little bit as anti-epileptics and also as analgesics too, to stop pain. Let's see, have I missed any other ones? Oh, opioids. Opioids bind to the same receptors that I was talking about with endorphin, enkephalins, and dynorphins. Opioids can bind to these receptors. So that includes morphine, codeine, hydrocodone, oxycodone, and the metabolic product of some of those is hydromorphone, which is more potent than morphine itself, and can agonize these to promote potassium efflux and inhibit calcium influx through those more natural opioids. Also, you have the phenylpyridines like fentanyl, remifentanil, and like meroperidine, which is kind of weird because it actually dilates your eyes instead of constricts them, but it has these other toxic problems, and it also can cause serotonin syndrome if you have too much of it, and also isn't very good if you have a problem with your kidneys. So anyway, I say all that to say that there's other classes. Fentanyl is more potent than morphine. It's a lot more. I think it's like 75 times as potent as morphine. And remifentanil is very short-lived, but it's used because it's short-lived. Like in surgery, if you want it to be short-lived and you want to actually time it exactly to the period of time that you're giving it, and you don't want it to keep lingering or lasting, remifentanil is a good one to use. But those are also opioid agonists. They would bind to this receptor. Let's see, did I miss any other ones? Oh, I didn't talk much about the NSAIDs. So NSAIDs are non-steroidal anti-inflammatory drugs, and those bind to cyclooxygenase 1 and 2, and those are fine. Those actually can help the periphery, not just the central part, but they also help the central, especially acetaminophen would bind, would only have its action in the central nervous system, but possibly by a slightly different mechanism. But the effect with NSAIDs, like naproxen, which is a leave, aspirin, which is acetylsalicylic acid or whatever, that binds to and inhibits COX-1 and COX-2, and you have diclofenac and ketorolac and indomethacin. Some of these drugs, they inhibit the production of prostaglandin E2, which I guess I'll draw. Prostaglandin E2. This is the big player. And NSAIDs, they block the production of this, and this is stimulatory towards like sodium channels, and the periphery is stimulatory towards I think it's stimulatory towards your transient receptor potential family V class 1 or whatever, the TRPV1 channel that senses pain, acid, and temperature, those things. It can also help sensitize that. So this is kind of mean, but it's also, you know, used in your immune system. So this is a major target of NSAIDs. This is what that's stopping mostly. But also they stop prostaglandin I2 as well, which I think causes vasodilation, if I remember right. Anyway, there's also drugs like, there's one for coughing that's I think the same thing as Robitussin, if I remember, but I might be wrong about. No, I'm wrong about that. But there's one that begins with a D and ends in orphan that I think that inhibits your NMDA receptors, but like in your C fibers, so you don't cough as much, because when you cough, that's actually being caused by C fiber excitation. And then there's also ketamine, which is a drug that does something similar to that. It affects NMDA receptors, but all over in the body. So it can cause you to like have hallucinations and stuff like that. But in your gray horn, for example, it's inhibiting NMDA. So the NMDA is not being excited by by the depolarization action here, and it's not letting in that calcium to signal CAMK2 to do the other activities in the cell that sensitize your cell. So ketamine is also being used to stop pain sensitization. So when you're on anesthesia, you might be unconscious, but it doesn't mean that these other processes don't occur, like sensitization. You're not knowing about it at the time, but you still have signal transduction in your gray horn, for example, and probably in your periphery. You have that stuff going on, you're just not aware of it, but when you wake up out of the surgery, that your sensitization processes still happen if you don't block it. And so opioids can block that too, and some people are administering opioids before, and some of maybe the drugs that block the calcium channels so you don't feel it, or so that you don't get the sensitization going on, so that when you wake up you don't have that problem. So I think I've covered most of the drugs that I've learned. I'm just not sure if I've missed any or not. That's pretty much it. So yeah, I hope you like this. If you like it, go ahead and share it.